Sorry, my computer's all weird. Stop it. Oh, sorry, did it start? Okay. For number 1, A, which of the terms describes or is an example of a regulatory protein, activator, and cap? B, which of the terms describes or is an example of a small effector molecule? We have inducer, allolactose, CAMP. C, which of the terms describes or is an example of a sequence found within the DNA? We have operator, -10, +1, LAC-Z, and LAC-A. D, which is not involved in lac operon, we have inhibitor. And structural genes are LAC-Z and LAC-A.
Hold on, it might have stopped. Oh, now I can't hear you.
Sorry, I made it. Mine still has a live in the box.
Since says hers is stopped.
Huh.
It's because she's the hoster. So.
Okay. I have like full Wi-Fi here. This video keeps cutting out. It's weird.
Oh, maybe we're still good.
I think we're good.
Sorry. Okay, we're good.
Okay.
So go exit out of it and then go back into it. Okay. So I'm doing number 2. The first one says, so A says, the lac operon is regulated by 2 inducers. Can you hear me? Oh, okay. Allolactose and CAMP. B, the binding site for sigma factor and the RNA polymerase is called LAC-P, while the CAP or CAP site is the binding site for the activator that regulates the lac operon. The enzyme adenylcyclase is what converts the ATP into the CAMP. And then D, it says the -35, -10, and +1 sequences are found within the LAC. And we got P portion of the LAC operon. And then E, we got the LAC-L gene encodes the LAC repressor, while the LAC-Y gene encodes the lactose permease. And then F, we got that attenuation can regulate gene expression in bacteria by causing transcription to terminate prematurely. And then G, so the last one we got, the E. coli strain that contains 2 copies of the lac operon is called, and we got a merozygote strain.
So for number 3, we're talking about an experiment that we have done that includes XGAL and IPTG in the growth medium, and how IPTG mimics allolactase, so on. So for the first one, we're trying to figure out, with all of these that are present or absent, if the culture will be blue, if the cells will be blue and metabolized. So the first one, IPTG is absent, glucose is absent, but XGAL is present. It's not blue because we need the other 2 for it to be blue. And then for B, they're all present, so it is going to be blue. C, IPTG is present, glucose is absent, XGAL is absent. So that would not work because we definitely need the XGAL for it to be blue, and we also need the glucose. D, they are all present. But with glucose, we don't want that for it to be blue. So that's not going to be blue. And then E with IPTG present and glucose is present, but XGAL is absent, blue, because we need XGAL to be there to be blue.
Okay. So for 4, we want to know if the gene is going to be inducible, non-inducible, or constitutive. So for A, everything's working as it should be, so it's going to be inducible. For B, it's non-inducible because the repressor can't be released from the operator because of that IS at the beginning. For C, it's constitutive. It never shuts off because the repressor can't bind to it. It's L plus is still on the chromosome, still works properly. The other on the plasmid is inhibited by the repressor and won't unbind to turn on. But because the chromosome was working properly, we decided it was inducible. And for E, we put constitutive, never turns off, and the plasmid, or the chromosome never turns on, and the plasmid never turns off.
Them have different variations of the genes and whether they're active or not, and then the lactose and the non-lactose. So trying to deduce how much yellow color tubes if both genes of the F plasmid and the chromosome genes both produce, there's going to be 2 yellow colors. Produces a yellow gene, or yellow, or only one produces proteins, then there's going to be 1 yellow color. And if they both don't produce anything, there's going to be 0 yellow color. So I think for the first one, only have so that and the doesn't work, so we're getting 0 on lactose positive and lactose negative. We got lactose positive sequence off. Lactose negative.